So good morning or good afternoon, depending on your location. So my name is Jérôme Guicheux. I'm from the University of Nantes and INSERM in France, and I'm a member of the Translational Research Committee of our lovely society, ICRS. That is my pleasure today to chair this ICRS webinar dedicated to cell therapy for cartilage with my wonderful colleague, Professor Aditola Aditya from the University of Alberta, Canada. Today, we are very pleased to have two internationally recognized experts in the field, Professor Nidhi Bhutani and Professor Ivan Martin. Thank you for having accepted our invitation. For everyone participating, please feel free to submit your questions using the Q&A button, and we will address them after the second presentations. Eddie, I let you introduce our first speaker. Thank you so much.
Thank you, Jerome. So Dr. Nidhi Bhutani is an associate professor in the Department of Orthopedic Surgery at Stanford University. Her research interests encompass the molecular mechanisms regulating regeneration and repair of skeletal tissues. Her research has identified a key role of epigenetic regulation through DNA methylation and hydroxymethylation in cartilage homeostasis and osteoarthritis. By using induced pluripotent stem cells and single-cell-based approaches, a multidisciplinary group has identified novel regenerative and pathogenic subpopulations that drive regeneration or degeneration in skeletal tissues. Dr. Bhutani has received the Arthritis National Research Foundation Research Scholar Award, the American Federation for Aging Research Scholar Award, and Innovative Research Grant from Arthritis Foundation. Her research is funded by the National Institute of Arthritis and Musculoskeletal and Skin Diseases, NIAMS, and the Department of Defense. So without further ado, Dr. Bhutani, please give your talk, Epigenetic Regulation of Cartilage Regeneration and Osteoarthritis. The floor is yours.
Maybe Nidhi has been disconnected, or?
I'm not sure.
Yes, she has been disconnected. One second, I'm going to try to see.
Okay.
Sure. Do you want to do that your own?
Yes, she asked us, can we start with Ivan? Because she's not on the call anymore. So Ivan, is it a problem for you to start?
No, it's not a problem.
So let me introduce Ivan. Professor Ivan Martin was initially trained as a biomedical engineer at the University of Genova in Italy, where he obtained his PhD in 1996. He was then a postdoc at Harvard MIT, and in 1999, he joined the Department of Biomedicine at the University Hospital of Basel in Switzerland as the leader of the Tissue Engineering Research Group, and is now a professor for tissue engineering at the University of Basel, and from 2021, is the director of the Department of Biomedicine. So even the floor and the mic are yours, even if Nidhi is coming back. So what are we doing? Up to you.
Okay, welcome back, Nidhi.
I'll start.
I think. So even. So maybe we can start with the event. So with the idea not to be too late at the end of the session. So
yeah.
Ivan, if it's okay, you can start if you are able to share your screen, or because we
Replace current share.
yeah, perfect.
Okay,
Yeah, okay. Thank
No
you so
problem.
much.
good. So glad to see that we are flexible, improvising to reactive to changes. So I'm not giving any introduction because I expect the audience would be better prepared than I am on the needs of cartilage regeneration and the connection between experimental models and the engineering of clinical grafts. And so I would like to directly dive into the product, into the solution that we have developed over the past years and that we have progressively clinically translated. So this is the NTEC. So it's a engineered tissue engineered cartilage out of cells which are taken from a nasal septum biopsy. Cells are first expanded, seeded onto the chondroguide membrane to generate patches of engineered cartilage which biochemically, biomechanically approach the properties of hyaline and native cartilage. Why have we been using a nasal chondrocytes? Because through the years, and now it's 25 years, we have not found a better source of cells that can generate so reproducibly in such good quality hyaline cartilage. And this has opened, thanks also to the embedding of the procedures into quality management system for GMP production, to first clinical implementation of this engineered functional cartilage. And this was initially for the reconstruction of a nasal alar lobule with full aesthetic, structural, and functional satisfaction of the patients. And now we have progressed recently to the reconstruction of the nasal septum using this cartilage. And the next step will be to develop a product for the treatment of empty nose syndrome. So in, let's say, cranial cartilage defects. But the largest clinical need is actually to use this cartilage towards joint cartilage regeneration. And after demonstrating the biological and biomechanical compatibility of this cartilage, which is generated from nasal cells into an articular environment, and this was more than 10 years ago, so do not go into that, we have had started the first in human implementation of nasal cartilage into the knee, initially for focal confined cartilage defects. And since then, so again, we are about 10 years ago, I can give an update on the clinical experience of this solution. So these were the results of the first patients. And as you see here, this was the KOOS self-assessed scoring system with regard to return to sport, but many other subcategories are equally, let's say, improving. We have a massive improvement in patients, and there was only this dotted line of a patient that returned too quickly to sport, so had an unrelated injury by playing soccer. And the return to sport was also confirmed by return to half marathon of some of the patients. Now, this is all anecdotal because this was only a pilot cohort. And since then, we have also started to extend into different joints and to start more in-depth studies for the knee. So just to give a bit of a time axis, we have now treated more than 100 cases for knee articular cartilage defects since 2012. We have started the first ankle and shoulder treatments, and we plan in 2025 the first treatment of the elbow cartilage. The NTEC, so this engineered cartilage, is currently under production for this first patient. And what we have asked ourselves, and what we are trying still to understand, is what is special and distinct in this nasal tissue engineered cartilage. And as the word says, first it's nasal. So we have a source of cells that, as depicted in this picture, comes from the cranial environment. So these are neural crest-derived cells that, as for many other neural crest cell compartments, have enhanced regenerative capacity, environmental plasticity. And we're trying to dissect now, in the context of a ERC Synergy funded consortium, what are the epigenetic signatures of these cells which endow them of this very special regenerative capacity. But this is a key distinct feature. The second is that we are not implanting the cells as a suspension or freshly loaded on a material, but we're effectively engineering a mature tissue. So we do not have, as written here, a CAM, so cell-associated matrix, but we have a TEC, so tissue-engineered cartilage. Does this make clinically a difference? And so we decided to start a phase 2 clinical study precisely addressing this question of whether there is a clinical outcome difference by implanting immature versus mature nasal tissue-engineered cartilage. And this was done in 5 clinical centers in 4 different countries, involving a total of 93 patients. And I'm happy to present these results, which were published last week. And first, related to the self-assessed scores. So here it's a bit of a challenging representation of the data, but the pink vertical line means that there is basically no difference between the treatment with NCAM, with the immature, versus the NTEC, the mature tissue-engineered cartilage. And the shift to the right implies that there is an improvement, which is in this case statistically significant, by implantation of the mature engineered cartilage. And what is also remarkable is that also for not only for the entire cohort, but also for the revision surgeries and for the larger defects, so that for the critical cases, we also have an improvement in implanting the engineered mature cartilage. These are subjective scores, and these are now instead objective magnetic resonance imaging derived data related to the classical T1 and delta R1, again indicating a superior clinical outcome in the case of a treatment with a more mature cartilage, not only at the site of the repair tissue where the engineered cartilage was implanted, but also in the surrounding cartilage, which indicates a possible beneficial effect of a graft to the other tissues in the joint. Now, if you're interested into more in-depth analysis of this clinical study, you may go into the paper that was again published last week. The good thing is that it was caught quite well by the public press. So as you see, this dated Friday, March 7th, so it's less than a week ago. What to me is extremely interesting of the work is the fact that the percentage of responders to this treatment in the entire cohort was very high, was close to 90%. But again, as mentioned before, also in the critical cases of the patients who were receiving a revision surgery or had large lesions, this percentage of the responders were equally, if not even higher. So this opens the space and the field clearly to the possibility that we can target even more challenging indications and maybe even use this procedure for degenerative pathologies like osteoarthritis. And it was 2021 when we offered the first biological basis for the possibility of treating osteoarthritic defects with nasal engineered cartilage. And this was based on preclinical in vitro and in vivo, even large size animal models, indicating that these grafts were capable of resisting to inflammation and even counteracting inflammation in the joint by reducing Wnt signaling through the SFPR1 protein. So we also had a postulated mode of action for this engineered cartilage, out of which we had permission from the Swiss medics, so the Swiss regulatory agency, to treat the first pilot cohort of patients with advanced osteoarthritis. And these patients now, who are some of them 5 years.
Network नहीं है.
Hello? Yeah. So, and these patients who are even 5 years after the treatment still have not, are living a normal life without any prosthetic implant, which would have been the alternative treatment for these cases of advanced osteoarthritis. Again, this is anecdotal, and we need to have a well-designed, fully powered study for this. And so we first decided to restrict the indication to a specific form of osteoarthritis, which is the one involving the defects in femur and patella. Sometimes these are called kissing lesions. And for these patients, which are relatively more uniform as the broad category of osteoarthritis, we have developed a technique that following tracheoplasty and debridement enables the implantation of two actually patches of engineered cartilage, one on the femur, one on the patella, in the two opposing lesions. And based on this indication, we have received generous funding from the Swiss National Science Foundation, from the European Commission, to treat a total of 225 patients. So the first 13 have been treated, but we are clearly at the beginning of the studies, and I will be happy maybe in a couple of years to give you an update on how this study is going. This is just the perspective for the future. But if we are entering this field of osteoarthritis and we want to control the disease, we also need to better understand it. And you all know that modeling osteoarthritis in a human setting is a great challenge. And animal models often do not recapitulate some of the biological features of human osteoarthritis. And why? Because it's a very complex disease, which starts from an abnormal loading and then through a variety of vicious cycles that finally ends to an inflammatory status, which is hard to control. And so we decided then to first try to develop models, maybe based on the buzzword of these years, so organoids, cartilage organoids, osteoarthritic human cartilage organoids, so that we can better understand the disease and maybe develop possible therapeutic modalities in combination, possibly, with our engineered cartilage. And so for this, I would like to present two possible solutions. So the first is based on the generation of these cartilaginous pellets or spheroids or organoids, which at some point during culture are exposed to inflammatory cytokines. And actually from the nice picture of the cartilage, we generate certain profile that is somewhat typical of osteoarthritis, with these foci of mineralization and a catabolic status in the organoid. And it's known, it's expected, but it was verified that if we treat with an anti-inflammatory agent, so for example, an antagonist of the IL-1 receptor, so we reduce this catabolic profile. And then through a proteomic analysis, thanks to mass spectrometry, we identified that the effect of IL-1 receptor antagonist was, among others, also driven by the inhibition of this transcription factor CEBP beta. And knowing that this transcription factor needs to be activated by certain kinases, we postulated that targeting and inhibiting these kinases would have similar effect as IL-1 receptor antagonists, which was verified experimentally. So this somehow validates the model and indicates how it could be used to define new pharmacological targets for disease modification, and that it could also offer some biological insights into the understanding of a disease. But ultimately, and this is my last part of my presentation, the osteoarthritis, as mentioned initially, starts from an abnormal loading, mechanical loading of a joint. And so this brought us to the hypothesis that by applying hyperphysiological compression into engineered cartilage in a microfluidic system, in a cartilage on a chip model, would induce certain traits of osteoarthritis. And this was a study performed in collaboration with a group of Marco Rasponi and Paolo Cheta in Milan. And here, the model is relatively simple because we first generate in these microfluidic wells human cartilage, and we are very good at doing this. And then we apply these levels of hyperphysiological loading, which induce certain traits of catabolism and inflammation. And then in a third step, we can start testing and screening possible candidate drugs into this model. And we see some differential response depending on the drug and compound, which indicates some form of possible specificity of a system. Now, before moving forward and deeper into this, we, however, considered that osteoarthritis is not a disease of the cartilage, it's a disease of the joint. And so we were pushed, especially by the reviewers, but also by the state of the art in the scientific community, to develop a mimic not of the osteoarthritic cartilage, but of the osteoarthritic osteochondral unit. And so we tried to make a bilayer osteochondral unit on this same microfluidic chip, and using articular chondrocytes and bone marrow stromal cells to generate these double compartments. And this also biologically, not only on experimentally and not only on paper, effectively generates two compartments, one with a let's say, calcifying marrow stromal cells, and the other with chondrogenic articular chondrocytes. Now, I would like to give only a couple of highlights on the beauty of the model, because you see that by using only articular chondrocytes or only bone marrow stromal cells, sometimes you have downregulation, this is the green color, of certain factors, in this case, anabolic factors. But the combination of the two cells under exposure of hyperphysiological loading gives an opposite trend. So it demonstrates the physiological relevance of having the co-cultured cell types under loading. And the same is in other areas. So for example, some catabolic factors that are upregulated in the presence of hyperphysiological loading, in the presence of a two single source of cells, but it's actually downregulated if these cells are co-cultured. And these are not artifacts, so it took us a long time to experimentally validate and confirm these data, which led us to the interest to look more specifically at the single cell level, what is happening upon hyperphysiological loading in this osteochondral on chip model. And we identified that there is one cluster of cells, which is here cluster 5, which only shows up if the two cell types are co-cultured and are exposed to hyperphysiological loading. So we were intrigued at what these cells are. These are still unpublished data. And we identified that indeed upon hyperphysiological loading, we see dysregulation of many of the mechanical actuator proteins, but also we were intrigued by a certain unexpected cluster, this one in red, of ribosomal proteins, which we were not aware, but we found in the literature in patient data to be associated to some recently identified dysfunctions of ribosomes in osteoarthritis. So this somewhat gave us the evidence that the system can capture some traits of patients that have never been identified in other in vitro systems. and that have never been associated directly with hyperphysiological loading. And so again, we very much trust that this system will enable us to better understand osteoarthritis and to better control it, possibly leading to a combination of the use of our engineered nasal cartilage with some combined pharmacological treatment. So I'm at the end of my presentation. I see that I'm at minute 20, so I'm glad that this is going about in time. So just to summarize, I started from the clinical experience in the implantation of nasal engineered cartilage, and then moving into the models, first the osteoarthritic cartilage organoids, and then the hyperphysiological microfluidic system, and now osteoarthritic cartilage on osteochondral, cartilage on chip that hopefully will be combined to better tackle this degenerative disease. In the pipeline, and just as a highlight, as a spoiler for the future, we have the engineering of osteochondral nasal grafts that could be used in certain specific indications, especially for the shoulder defects, and also a bit of a blue sky vision, but the possibility of having nasal chondrocytes, which are through synthetic biology approaches sensing certain pathological, environmental, osteoarthritic cues and can functionally rewire these signals into regenerative signals as designer cells. So this is a subject of new grants that we hope will be granted and will enable us to work on this. So with this, I would like to acknowledge funding collaborators, especially the group of Andrea Barbero, with whom we tightly interact. And this evening or in the lunch break, remember that you may try this position of the yoga, Janu Sirsasana. The head goes to the knee, the nose can help the knee. Thank you very much.
Thank you, Ivan. Thank you so much for this inspiring talk. I think now it's time to give the mic to Nidhi for the second talk of this webinar. Thank you again, Ivan.
Thanks, Ivan.
Hi, everyone. Can you hear me and see the slides?
Yes.
Okay, perfect. Yeah. Hello, everyone, and hope you're having a good day since we are all spread out across multiple time zones. And thank you for that fantastic talk, Ivan. And I will shift gears a little bit and try to talk about epigenetic regulation of cartilage regeneration and osteoarthritis. So again, as many of the people on this call know, that cartilage is a tissue which is difficult to repair. And the reason for this is that there is an intrinsic, a low regeneration potential, which is likely because of the low cellularity of this tissue. This tissue is avascular, so the access to stem cells is somewhat limited. And really the question then is, what are the cartilage intrinsic stem cells or regenerative populations that we can identify and potentially harness for cartilage repair? So even give a beautiful talk about using nose cartilage cells to repair knee cartilage cells. So the question here then is, are there regenerative populations in the endogenous positions in the cartilage, which we can probably try to understand more and think about other biological factors to supplement tissue engineering approaches, or to try and devise approaches where we can modulate these endogenous stem cells to enhance cartilage regeneration. And of course, this is because cartilage injuries lead to a huge unmet medical need of osteoarthritis, and the economic burden exceeds 50 billion per year. So osteoarthritis, again, as we all know, is an age-associated disease which has no effective disease-modifying drug available. And a part of the issue is that the early events in OA pathogenesis are still elusive. And despite multiple GWAS studies, there are no unifying genetic loci which are associated with OA. And it is a progressive degradation of the joints where the disease really manifests itself through degeneration of cartilage as well as bone. And because there was no unifying genetic loci, we turned our ideas and hypotheses towards epigenetics to try and sort of understand how are epigenetic changes taking place during osteoarthritis, and whether that is something which can help us understand how to tackle this therapeutically. So epigenetics, of course, is a huge wide field, and we have sort of focused our studies on DNA methylation. But of course, when you think about epigenetic regulation, it involves histone modifiers, chromatin remodelers, as well as multiple factors which can really affect chromatin accessibility and gene expression as an end. There's also a huge word of regulatory RNAs. But our studies, we have sort of really focused on this covalent modification of DNA methylation. So when we think about a DNA methylation, it's basically carried out by this group of enzymes, DNA methyltransferases, which add this methyl cytosine group, methyl group onto the cytosine, resulting in this mark 5MC, which traditionally is associated with gene repression. So the presence of 5MC in regulatory regions like promoters and enhancers is conventionally associated with gene repression. And until around 2010, our understanding was that this mark is quite stable. It is something which is added on to cytosines during development and sort of stays as is. But then during that time, our understanding of this epigenetic mark really changed quite dramatically, because the discovery of TET proteins, 1011 translocation proteins, revealed that this methylation mark can actually be modified further. It can be hydroxymethylated into this mark 5-HMC, which can be quite stable. It is present in the DNA, and it's actually associated with gene activation, especially when found in gene bodies. So this can be a stable mark, or it can also be a transient intermediate towards DNA methylation, as it can be oxidized further to basically lead to an unmethylated DNA, unmethylated cytosine. So, so this is an intermediate in DNA demethylation as well as a stable mark. But what it really means is that this DNA methylation is a very, very dynamic mark, which can change during disease. And hence, we really have to understand this DNA demethylation dynamics to sort of see how the effect of this whole cycle is on a cellular state. And as I'll tell you further, this plays a big role in osteoarthritis as well. So when we started looking at DNA methylation and hydroxymethylation, it was already known that there are genome-wide alterations in DNA methylation patterns in OA cartilage. And what we discovered was that there was also a huge aberrant DNA hydroxymethylation in osteoarthritic cartilage. What we found was when we compared OA cartilage with normal cartilage, and this OA cartilage is from patients undergoing total knee replacement, we found that there was a huge increase in the levels of 5-HMC in these osteoarthritic chondrocytes. We went ahead and did a genome-wide mapping of this 5-HMC. And again, we saw that this increase was quite significant, and it was really there in regulatory regions, like in promoters and enhancers, as well as, interestingly, there was a huge increase in 5-HMC levels in the gene bodies of these cartilage genes. And this again sort of shows that it was in gene body that this enhancement of 5-HMC was very, very prominent. So when we looked at the different pathways which were associated with this enhanced 5-HMC, what we saw was that a lot of OA-associated gene pathways were showing up. So this increased 5-HMC in the promoter regions was associated with Notch signaling, Hedgehog signaling, and the increased 5-HMC in the gene bodies was associated with Wnt, pathways of cartilage development, FGF. So a lot of these pathways which we know are playing a regulatory role in osteoarthritis. Interestingly, we saw very clearly that this increase in 5-HMC was there only in genes which were activated in OA. So as you can sort of see here, when we looked at genes which were increased in osteoarthritic cartilage, we see this 5-HMC enhancement in the promoter regions as well as in the gene body. But when we look at genes which were either decreased in OA or genes which were not changed in the osteoarthritic cartilage, we didn't really see this 5-HMC enhancement. So this sort of showed us that this is correlated with OA gene activation. So then all this work was in human patient samples, and we went back to an OA model of osteoarthritis to sort of try and see whether this increase is observed there as well. So for this, we used a well-established model of osteoarthritis, the destabilization of medial meniscus. And here, after surgical injection, by 6 weeks, we start to see early OA, and by 12 weeks, a full-fledged OA is observed. And we looked early on to see whether this increase in 5-HMC is associated with early events in OA. And this is what we observed, that yes, indeed, the mouse model also recapitulates this global increase in 5-HMC. This here is 5-HMC staining in the DMM model as compared to the sham with a specific 5-HMC antibody in the cartilage. And you see that there is an increase in 5-HMC, which is quite significant. So then the logical question was that, so is this increase in 5-HMC regulatory? So this means that if we can inhibit it, or we can use a TET1 mutant mouse where this 5-HMC increase would probably not happen, what would be the effect on osteoarthritis? And so before this, we did 5-HMC sequencing again, and we observed that in the wild type situation, not in the mutant, we saw that there was again this enhancement in 5-HMC. It was really upregulated in multiple sites. And again, this increase in 5-HMC was at the genomic locations that we saw in the human patient samples, that is in the promoter and the gene body. There was a very good overlap between the human and mouse data. A lot of genes which were upregulated in mouse were also upregulated in human, but there were also differences, as we always see. When we look at the pathways, again, Wnt and FAT, these were common pathways which were associated with the increase in 5-HMC. So then as I said, we went back to a mutant mouse, and for this we used the TET1 mutant. So TET family has, there are 3 enzymes, TET1, 2, and 3, that we just sort of started with the TET1 mutant. And what we saw was very interesting. What we saw was that in the TET1 mutant, basically the mice were really protected from osteoarthritis, which was induced by surgery. So here you can sort of see this is the wild type, where after 6 weeks after the surgery, you see there is establishment of osteoarthritis. We see cartilage degeneration, fissures, as is expected. But looking at the mutant mouse, we really saw that the cartilage was quite pristine, and there was no telltale signs of OA in these mutant mice. When we look at MMP13 expression, again, we see quite pronounced expression at 6 weeks in the wild type mice, but in the mutant mice, we didn't really see any expression. So upon doing ORC scoring, we could see that there was an increase in ORC score in the DMM model with the mouse, with the wild type mouse, but in the TET1 mouse, really between the sham and DMM, there was hardly any increase in the OA score. Similar with the loss of cartilage, we saw that the loss of cartilage in the TET1 mutant mouse was really very minimal. So when we looked at the 5-HMC, we saw, interestingly, that most of the 5-HMC gain that we see in the wild type is not observed in the TET1 mutant mouse. So this actually told us that TET1 is the major enzyme which is involved in this 5-HMC deposition in osteoarthritis, and not TET2 or TET3. So we just, so it looks like this was the right mouse model to pick for this experiment, because TET1 seems to be the major enzyme which is involved in osteoarthritis. Then, of course, we went ahead and did RNA-seq to understand what are the genes which are affected or are actually not upregulated in this mutant mouse. And what we saw was that in this mutant mouse, really Wnt signaling was dampened, p38 signaling, senescent cells-associated SAS proteins were dampened, and there was an increase in some chondroprotective genes, as well as there was an increase in N-glycan biosynthesis. So this really gave us a nice snapshot of pathways which are important for early OA and are 5-HMC dependent. And in the absence of TET1, we basically did not see this upregulation of OA-associated genes in cartilage, suggesting that this is why the cartilage is protected in this mutant mouse. Then we also did another experiment. We asked this question that if there is actually inflammation would this cartilage be protected if it does not have TET1? And what we saw was that when we injected IL-1 beta in the knees of wild-type mice or the mutant mice for 6 days and analyzed cartilage at the end, in the wild-type mice, as expected, we saw that there was a loss of GAG, there was a loss of these proteoglycans, which was very prominent. But in the TET1 mouse, there was really no loss of GAG that was observed at all. When we looked at MMP13 expression again, MMP13 was not upregulated in the mutant mouse while it was upregulated in the wild-type mouse. So what this told us was that this loss of TET1 really renders an intrinsic protection to cartilage. That even if there is inflammation around this cartilage is protected if there is no TET1 and there is no TET1-mediated 5-HMC upregulation. We went ahead and did this again in vitro using an ATTC5 cell line where we used a shRNA-mediated knockdown of TET1, and we pretty much saw the same thing, that when TET1 is not there, we don't see an upregulation of MMP13, 9, or IL-6 when this cartilage is treated with IL-1 beta, again showing that there is this intrinsic protection to inflammation when there is no TET1. Then we went ahead and tried to ask this question of whether now we can also modulate end-stage OA chondrocytes from patient samples with TET1 loss. And again, we see that when we were able to downregulate TET1 using a shRNA-mediated approach in these OA chondrocytes, we saw again that there was protection. There was a loss of inflammatory markers like CCL2, WEAK1, VEGF1 in these OA chondrocytes, as well as there was a downregulation of MMP3 and MMP13. So this showed that TET1 inhibition can actually be therapeutic because this is now end-stage disease that we are talking about. So then we went back and tried to see if we can use a small molecule inhibitor of TET1 and whether that would also be therapeutic in this mouse model of OA. And we found that the answer was yes. After surgical induction, in this case, we injected a small molecule inhibitor of TET1 to HG, which is a broad inhibitor of all TET proteins, as well as some other enzymes. And these injections were done twice in a week. And after 6 weeks, we saw that this inhibitor was again able to give the same protection that we saw in the mutant mouse. So this is the control and inhibitor, and you can see that there is substantial protection from cartilage degradation. Again, the ORC scores were modified. MMP13 was reduced with the inhibitor. And importantly, we saw that when we looked at the 5-HMC levels, we saw that with the inhibitor, the 5-HMC levels were down as well. So this means that really it is the deposition of 5-HMC which is responsible for all the effects that we see of the inhibitor. So this was sort of like a protection, right? Because we started the inhibition very early on, right after the surgery. So then we asked this question of whether this would also be therapeutic when the disease is already set in. So our data from the human patient samples suggested that this would be the case, and we wanted to test it in the OA mouse model as well. So in this case, after surgery, the mice were left for 4 weeks so that the disease is already initiated, and that's when the TET1 inhibitor was introduced. And what we saw was, again, that TET1 inhibitor was therapeutic in this case as well. We saw that the ORC disease scores went down. Interestingly, we saw that there was also, of course, the percentage loss of cartilage was significantly downregulated, as you can sort of see here as well with the 2HG. But what we saw was that there was actually an enhanced cartilage regeneration. So, sorry, I'll come back to this. So when we look at it more closely, what we saw was that with the 2HG, we actually saw an increase in cartilage regeneration. When you look at cartilage loss at 4 weeks, we have around 20% loss. But with the 2HG, it was not just that we recaptured this 20% loss or we inhibited any further loss. There was actually more cartilage growth. So this was interesting. So this meant that we could probably get an idea about how cartilage regeneration is happening in this mouse model. So then we turned our attention to skeletal stem cells and tried to understand whether skeletal stem cells are really responsible for this enhancement of cartilage regeneration in this model. And for this, we collaborated with Chuck Chan's group, who had discovered the whole skeletal stem cell lineage. And just to sort of run you through the skeletal stem cell lineage, so the skeletal stem cells are supposed to give rise to these bone, cartilage, stromal common precursor, which can then differentiate into stroma, into bone, or in cartilage. So this is the chondrocyte precursor cells that we were very interested in. So for these experiments, Akshay Pandey, a very talented postdoc in the lab, he basically took the TET1 wild type and mutant mice and isolated skeletal stem cells and the remaining lineages from these. So for this, he used P3 to P7 pups. These are very, very young, just in the first week after birth, where the skeletal stem cells are at their highest frequency. The skeletal stem cells do go down with age, and that's why we sort of started with very, very young pups. And after a mechanical and collagenase digestion of their joints and staining for SSC lineage markers, he was able to sort these different populations. And interestingly, what he saw was that in the knockout mice, the percentage of the skeletal stem cells was already significantly higher as compared to the wild type. So, so it looks like the SSCs were much more in number, and as a result, they did not differentiate so much into the further lineage. So you saw a decrease in the further lineages like BCSPs, chondrocyte precursors. They were all down in frequencies because the SSCs seemed to be a little bit higher. And so this means that maybe these skeletal stem cells are able to self-renew a little bit more. But the second question we asked was that if we were to now isolate these SSCs and subject them to chondrogenic differentiation or osteogenic differentiation, is there a difference in their propensity to differentiate as well? So for this, we took the wild type SSCs and BCSPs and subjected them to chondrogenesis. This takes around 3 weeks with the required, with the relevant growth factors. And what we saw was that not only the SSCs were higher in the knockout, these SSCs also differentiated more towards chondrogenesis. So they had a skew towards enhanced chondrogenesis in the absence of TET1, which was very, very interesting. So this is just from three different mice. So in the replicates, you can see very well that the TET1 knockout mice actually have a higher tendency towards chondrogenesis. And this was not observed with osteogenesis in the knockout and the wild type. The tendency to differentiate towards bone was very similar. And this is just quantitated here. You can see that the chondrogenic markers of differentiation called 2A1, called 1A1, as well as SOX9 were much higher in gene expression in the knockout SSCs as well as in the BCSPs. So this was, as I told you, in very young pups. So then we went back to 3-month-old mice, which are more like adult mice, and asked whether that was the case in these mice as well. So here the frequency of SSCs and BCSPs is lower than in the pups, but we saw that the increase in SSCs was still maintained here, and the rest of the lineages were low. And then we asked this question of what about if we injured the mice? Is there an increase in these skeletal stem cells? And here the mouse was injured, and we looked at it after day 7. And what we observed was there was a slight increase in SSCs after injury in the wild type, but it was much more significant and more pronounced in the knockout mouse. So in the knockout mouse, after injury, there was an increase in SSCs, which as well as an increase in BCSPs, and then they were also skewing towards enhanced chondrogenesis. So this is again with the injured BCSPs as well. We saw that the skew in chondrogenesis was maintained. So then we went back to human samples. We isolated human skeletal stem cells from OA cartilage as well as chondroprogenitor cells, and we saw that the increased, the skew towards chondrogenesis was there in the human samples as well. So this is the control treated and the human and the 2AG treated human SSCs and human chondroprogenitors, and we saw that in both the cases with the TET1 inhibitor, we could increase the chondrogenesis of SSCs as well as CPs. So this means that we can really use a 2AG or inhibition of TET1 as a biological modulator to increase the chondrogenesis in these stem cells. And then we really wanted to understand, like, what are the downstream factors which are leading to this enhanced chondrogenesis. So for this, we did RNA-seq in the wild type and the TET1 knockout mouse, and in the SSCs as well as BCSPs. So the first thing we noticed is that the pathways which were modulated here were very different from the pathways in the differentiated chondrocytes, in the articular chondrocytes that I had shown you before. Here, what we noticed was that a lot of metabolic pathways seemed to be perturbed, and one pathway which really jumped out at us was the melatonin degradation pathway. The melatonin degradation pathway was really reduced in SSCs as well as BCSPs, which would mean that they would have enhanced melatonin. So melatonin, as you all know, is a sleep hormone, which is FDA approved. It's available over the counter. So this was sort of a very interesting and exciting data for us. So then we asked this question of whether melatonin is really one of the consequences of TET1 loss, and whether that can mimic some of the effects that we see with the TET1 inhibition. And what we saw was that, as compared to DMSO, if the SSCs were supplemented with melatonin, we saw an increased chondrogenesis in SSCs as well as in BCSPs. So looked like melatonin was able to mimic the effects of TET1 inhibitor on SSCs and BCSPs. Then we went ahead and tested it on OA explants. So these are OA cartilage discs, which we could get from knee joints from patients that were undergoing total knee replacement. And when these OA explants were treated with control or the TET1 inhibitor or melatonin, we saw that melatonin could have a very similar effect to the TET1 inhibitor. There was enhanced cartilage regeneration and GAG secretion in these OA explants, and which was very comparable to TET1. We also looked at the cytokines which were secreted by these OA explants, and with the TET1 inhibitor, we always see inhibition of IL-6 as well as some other cytokines. CCL2 is another one of those. And what we saw was that with melatonin treatment, we again saw inhibition of these cytokines as well. So it does seem like melatonin is a downstream effector of TET1. So what we are able to sort of understand from all these studies is that these skeletal stem cells are involved in articular cartilage regeneration endogenously, and these data really provide us many strategies for modulation of endogenous stem cells in cartilage repair. These epigenetic regulators can probably provide us, therapeutic strategies for OA. The challenge, of course, with targeting these broad epigenetic regulators is that there can be off-target effects, and that's why it's important to understand downstream effectors of these epigenetic regulators. For example, like melatonin, which can possibly be used in clinic, as Martin was saying, with probably associating them with more tissue engineering strategies to use these biological modulators with the right stem cells to try and enhance cartilage regeneration in osteoarthritis. So I'll stop here and thank my lab members. The highlighted members really contributed to these studies, as well as our collaborators and funding agencies, and I'm happy to take any questions. I'll stop sharing my slides now.
Thanks very much, Nidhi. That was awesome. So questions here?
Maybe I can start with the first question for Evan, Eddie.
Okay, go for it.
Evan, I know that you have been interested in the use of nasal chondrocyte, not for the knee, but for the back. Can you tell us a few words regarding what you have done and where are you with this new field, nose to back?
So we are also testing the use of nasal chondrocytes, in this case, in the form of spheroids injected into intervertebral disc. After demonstrating that they can very well resist to the harsh environment of the intervertebral disc, so low oxygen, low glucose, high inflammation. And so far, we have demonstrated the capacity to engraft into chronically degenerated discs in ex vivo models and in large size animal models. And we are running a study on sheep to test the possible therapeutic effect. But so far, we realize what is typically a challenge for injected cells into the disc, that they can survive, they can engraft even long term. So we will see, but that's indeed the nose to back approach, which will not be simulatable in a yoga position.
Okay, okay. Thank you.
Nidhi, I wanted to ask you about the TET1 model that you were using, the mouse model. Were these exercise induced as well? Did you try the exercise induction as well?
Yeah, no, that's a great question, but no, we have just used either just the DMM model, or we have used the tibia loading model, where we actually load the tibia directly so that there's an ACL rupture to kind of mimic the ACL rupture in humans, but we have not really exercised them further to induce more degeneration.
Yeah, could you speculate what you might get with the exercise
Yeah.
induction?
So exercise is always a little tricky, right? Depending on the intensity of the exercise and when you actually start exercising them, it could be beneficial or deleterious. So we haven't really tried that in our models as yet. But I would speculate that, I think initially after, when the joint is unstable, what we have typically seen is exercise worsens the OA rather than help it. But I know that if you do it a little bit later, and also in humans, exercise is supposed to help. So it's tricky. I don't know if it answers your questions, but
it does,
it's complicated.
Actually. Yeah. It does.
Actually, may I also ask a question on this context? So in your mouse knockout model, when you that's a conditional knockout, I assume. So
no, so we've just used global knockout, and because we do have a specific inhibitor, so the inhibitor was injected intraarticularly to have a local effect.
Okay, so I wonder what are then the cells that mostly are affected in this knockout or silencing of TET1, because you showed the skeletal stem cells may have a role, but did you check other cell types? What would be regulating, whether they also would be regulated, including synovial membrane cells and inflammatory mediators?
Yeah, so that's a great question. So what we, I didn't show the data, but that data is in the paper. So one possibility was also just that the immune cells are not responding the same in this mouse model, right? Because it's a global knockout. So we did check for infection. So if you do a LPS modulation, for example, the wild type and the TET1 knockout behave the same. So it was not as if there was a global dysregulation of inflammation per se. And when we looked at synovitis, we do see synovitis is reduced, but because there's a crosstalk between synovium and cartilage, it's a chicken and egg situation, what comes first. So we don't think that it is really immune, that it's immune modulation, which is affected first in this model. And the experiment that we did, where we had we kind of induced inflammation in the mouse to see what would be the effect on cartilage. That tells us that really it is a cartilage intrinsic property. The cartilage is sort of more resistant to inflammation. So we do think that the first effect is on cartilage. And this regenerative effect, we haven't shown it directly. It's still an open question in the field, right? The skeletal stem cells, how do they actually get to cartilage to enhance this repair? But that is something we are trying to understand a little bit more. Again, it's a complicated situation, but we are trying to understand a little bit more.
But
what
it's
is
fascinating.
Yeah. Thank you.
Ivan, if I may ask you a question about your implant. So you talked about the nasal tissue engineered construct. When you implant it, it's actually, there's a kind of synergistic interaction between the surrounding cartilage as well, that there was some benefits to the surrounding cartilage when you implant it. Could you comment on the mechanisms that might be involved in that?
So I mentioned probably too quickly, but what we have investigated is the co-culture of these nasal chondrocytes and synovial membrane cells, both from osteoarthritic patients or from normal patients, but during exposure to exogenous inflammatory cytokines. And what we observed very clearly is that in the presence of the nasal chondrocytes, the synovial membrane cells were reducing their inflammatory profile. And when we looked at possible mediators of this, we identified that there was a inhibition of the Wnt signaling in synovial membrane cells, and that this could be mimicked by SFPR3 cytokine that is produced by nasal chondrocytes and not by articular chondrocytes. So this is probably just one of the different factors, but we recognize that there could also be a secretome of the nasal chondrocytes that positively influences the joint environment to reduce the inflammatory profile.
Fantastic. And if I may ask a follow-up question as well. So with the implants, you're putting them at different, biomechanically different environments as well. Shoulder, ankle, knee. Could you just kind of like speculate about the adaptation of the nasal tissue engineered construct in these microenvironments that are biomechanically different?
Yeah, so obviously I can refer to data generated in preclinical models, but in large size loaded animal models where we have implanted our nasal engineered cartilage after labeling the nasal chondrocytes. And upon implantation and exposure to normal loading and harvest of the cartilage, we could identify nasal chondrocytes still labeled as such that were forming the typical columnar structures of articular cartilage. So they clearly can respond to loading and generate these architectures that are mechanical loading associated. What are the mechanisms for that? We don't know. We also applied shear forces in vitro to these nasal chondrocytes, and we also found upregulation of lubricin, which is typically not present in nasal cartilage. So there are several signals indicating plasticity also to a mechanical and adaptation to a mechanical environment, which is not typical of the nose.
Thank you.
Yeah, I have a follow-up question for Evan as well. So I think your data shows that the younger chondrocytes seem to have a better adaptability or a better response, right, than the older chondrocytes. Do you have any sense of what biological factors might be modulating that?
So the younger versus the older nasal chondrocytes, or from we do not observe much age dependency in at least in the in vitro capacity of the nasal chondrocytes to proliferate and regenerate new cartilage. And this is much in contrast with the behavior of articular chondrocytes, which at least in our hands is very much age dependent.
Yeah. So, I mean, yeah, that's what I was wondering, that there is this age-dependent variation in the articular chondrocytes. So you don't really see that in the nasal chondrocytes. That's interesting.
We do not, no.
So there's a question.
Look at the epigenetic regulation.
Yes. Yeah, maybe they don't age so much, which is very interesting, actually. Yeah.
I've taken some notes of some of your candidates that you were showing, and we look into that.
We should get that. Thank you.
So we have a question in the chat from Jawadi Adi Raja from India. I think this is more of a question to possibly an orthopedic surgeon. Will correcting the alignment of knee before these procedures help in any way? I think this is more to you, Ivan.
So clearly, a correction of the axis of the long bones have a profound effect on the preservation of the cartilage. And indeed, not for patellofemoral osteoarthritis, that has a different origin, but for patients that we have treated with medial osteoarthritis. And this was a very small cohort as a first attempt to treat osteoarthritis with our engineered cartilage. We have done correction of the axis and implantation of the engineered cartilage. And so we have seen a massive improvement in the clinical outcome of the patients, but we cannot decouple the effect of alignment from that of the implantation of the cartilage. So that's a very important point.
So there's another question here.
Yes, I think there is another question for Ivan.
Yeah. Liam, do you want to take this one, Jerome?
So there is another question for you, Ivan, from Liam McKeon. As in human research, the membrane potential of nasal chondrocytes versus other cell sources as a possible mechanisms for why they retain greater regenerative capacity?
Not as far as I know. Maybe you could include in the chat or send me a message. Why do you think that the membrane potential especially could be implicated? This is intriguing. Maybe we should do the next experiments testing that.
Thank you. Thank you. I think if there is no more question in the chat, it's time to close the session because we are a bit late. So thank you so much, Nidhi, and thank you so much, Ivan, for being with us. I hope everyone you enjoyed this webinar, and we will be very much happy to see you for the next webinar of the ICRS. Thank you so much, Ivan. Thank you so much, Nidhi. Enjoy the rest of your day, and see you around.
Yeah,
Thank you
thank
very
you.
much for organizing it.
Thank you so much. Bye, Eddie.
Enjoyed it a lot. Thank you. Bye.
Thanks, Nidhi.
Bye-bye.
Bye-bye. Bye, Joe. That was awesome.
Bye, Eddie. See you.
Yeah, bye.